This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 30 for May 1st, 2009. From New York City, I'm Vincent Racaniello.
I'm Despommier.
I'm Hamish Young.
And in Western Massachusetts, I'm Alan Dove.
Today's an incredible day for virology, gentlemen.
Indeed.
Last week's episode, we just missed this swine flu.
We did.
I forgot to welcome Hamish back to TWiV. Hamish, welcome back to TWiV.
Thank you very much. Glad to be here.
Thanks for joining us. It's quite an episode to be on.
Yes, I was going to say, this is the crisis.
In fact, in all of my years of virology, about 30, and you more,
right.
This is probably the most interesting and exciting week we've had, because we're watching a virus spread in real time, right?
1999 wasn't so bad for a virus in New York either.
SARS. And then SARS,
West
too.
Nile.
West Nile. And then SARS, too.
Sure.
Wasn't exactly... I remember being much more worried about SARS.
Yeah, me too.
And we had, don't you remember, Vince, we had meetings of the whole community, got together with Hammer, people from the public health school, talking about SARS. Have you had a campus-wide meeting yet about swine flu?
Not
I
yet.
doubt it.
No.
SARS really got everybody very upset.
Well, SARS was a lot scarier in a number of ways.
Yeah, I mean, 50% fatality rate.
Yeah, and also no antivirals, all the things we're going to talk about here.
And we didn't understand the transmission cycle.
Right.
I'm not
Which
sure we
was
understand the transmission cycle here either, actually. But
Achoo.
All right. Well, I agree that we were quite excited about SARS, but that died out quickly.
yes.
And
But why is not terribly clear to me. And why it's disappeared off the radar screens completely.
We had a show on that, actually.
Yeah.
When we
it might come back, but
speculated about bats.
Here
and
we
stuff
have
like
swine flu, or a influenza virus that originated in pigs. And when we last had our TWiV last Friday, we had cases in Texas and California. And as we were talking, Mexican cases were confirmed to be caused by the same virus. And Alan, you saw the breaking news on the BBC.
Right.
And since then, over the weekend, it exploded. Cases have been reported everywhere. And in fact, let's review the current situation in the U.S. I think we should talk about laboratory-confirmed cases.
Mm-hmm.
Yes.
Absolutely.
Yes.
Meaning that the patient has had a sample taken, typically a nasal swab or a throat wash, and either virus is isolated and confirmed to be influenza H1N1, and some kind of antibody or serology is done. Of course, the cell culture taking much longer than or DNA methods, which are used to diagnose it. So that's a laboratory-confirmed diagnosis. In the U.S., we have 141 cases as of today, and one death, which was actually someone who
imported from Mexico.
Right. And very, very young, in fact.
And most of these are in New York, because we know that the kids at the school came back from Mexico.
Right.
So we have 141. They're scattered all over the U.S.
So there appears to be an epicenter of where this originated.
Well, let's say there are 28 in Texas. So those came from Mexico, I presume.
Yep.
There are 13 in
Southern California.
Southern
Mostly
California.
Southern California.
And then all the other states, Arizona, Colorado, Delaware, Illinois, 3, 2, 1. They're all over the place. New Jersey has 5. And this probably reflects people traveling
Correct.
from other infected areas.
That is the assumption.
Now, let's look internationally. We have a total of 11 countries, including the U.S., 331 confirmed cases and 9 deaths, basically 8 in Mexico and 1 in the U.S. Is that right?
Yes.
Let me check that.
Not 8 in Mexico. 8 confirmed in Mexico.
8 confirmed.
I see. Yes.
I saw 12 confirmed in Mexico.
Really?
At one point, but I don't know if that was inaccurate or newer.
Did you see a breakdown as the age group of those people?
No.
I have not.
Me neither. I'd be very curious to see that.
I'm looking at the WHO site, which is updated May 1st, and those are the numbers.
Right.
Okay.
11 countries, 331 cases. But as you know, there are many, unconfirmed or
That's right.
suspected
Inapparent
cases.
infections, of course.
And in fact, most of those will not be confirmed because you don't isolate virus or confirm viral RNA from everyone.
Right.
but you will have some sense of which are flu and which are not. So that's the current situation. And my sense is that it is still spreading quite rapidly. And this is basically a new virus against which we don't have antibodies, right?
So I have a question as a lay person in this field.
You're a virologist.
No, but lay in the sense of not knowing the intimate details of influenza virus.
Mm-hmm.
So here we have an H1N1 virus. Now my recollection is, back in 1976, when there was another swine flu outbreak in Fort Dix, that hemagglutinin used to be referred to as H-swine.
Right.
Now, was that H1 as well?
Okay, these are all H1s, yes.
They're all H1s. And the pandemic of 1918 was H1.
Yes. So, what would you like a brief history of
yes,
swine?
of H1 is what I'm
all right. So, the H1 of 1918, the human virus was an H1N1.
Right.
About the same time the pandemic began and it went into pigs.
Right.
And that virus remained in pigs for many years.
30-odd years or whatever.
More. In 1998, pigs became infected with an H3N2 strain.
From an avian source.
From human.
Oh, from human. Mm-hmm.
In Europe, pigs were infected with an H1N1 avian virus.
Right.
As well as other human viruses. And these have all recombined in pigs and evolved.
Right.
And so now the H1N1, it's difficult to know if it's a descendant of the original classical H1N1 or if it's the European avian-like. But basically, these viruses have all evolved as recombinants of human and pig and avian strains. But now, of course, they all look like pig viruses because they've been in pigs for so long. Right. So this is in part of the confusion. Last week, someone said this is a virus with genes from human strains, pig strains, and bird strains. But if you look at the sequences that have been released, and if you compare them to the sequence database, they all look like pig strains. And that's because, they are pig strains. They've been circulating in pigs for a dozen years, but their origin is from human and avian strains.
I see. Okay.
So another lay question. I ask only lay questions because I am a lay person. I'm curious to see what the comparison of the genome of the 1918 H1N1 is, because we have the entire sequence of that genome.
We do.
Compared to the H1N1 of today.
It's very different.
Exactly.
It's very different. It's not the same virus.
No, it is not the same virus. So even though we're talking about H1N1s in both cases, we should be very clear about the fact that today's H1N1 is much, much different than 1918.
Oh, of course.
Okay.
So
just wanted to get that out of the way right away.
One of the things that I found very interesting watching the news is how initially it was always referred to as swine.
Yeah, exactly.
And then all of a sudden, all the newscasters started to talk about H1N1, as though this would suddenly reassure people.
No, it was an edict from the NIH through Tony Fauci, who had actually made an announcement.
Oh, he did? Okay, so
and he said from now on, we're going to refer to it as flu because flu is flu is flu. This is a strain A flu, H1N1.
Okay.
It can infect pigs, it can infect people.
Okay.
Which I think was actually a pretty compelling rationale.
I thought so too.
Because it's not informative anymore to call it swine flu. Initially, it looked like maybe it was coming from pigs into people, and we were just seeing the beginning of this.
Yes.
But now it's clear that this is spreading person to person. I don't even think it's clear that pigs can catch it anymore.
That's right. I agree.
This is effectively a human virus.
So one of the tragedies of this misconception is the fact that Egypt slaughtered all of its pigs.
Yes, I heard that.
And that represents a huge protein loss for many, many, many people.
And a huge economic loss.
Economic loss too.
Yes. And my wife asked me, she said, why are they killing all the pigs in Egypt? And I said, well, because they're being hysterical. But I don't know if that was quite the right phrase, but I think it was.
That's spot on, yes.
Yeah. Well, the idea was that the virus was continuously coming from pigs into people, which you can understand a lot of people would think. If you call it a swine flu virus, they think, well, it's continuously coming from pigs, so let's get rid of them.
That's right.
In fact, it was once a swine flu virus, but as soon as it started passing among people,
means it's a human virus.
You selected now for a virus that was completely different. Whether or not this could go back into pigs, we don't know. It might, because human strains do infect pigs regularly. It could, but there is no reseeding of humans from pigs anymore. This was a chance event that generated this virus.
That's right.
A pig somewhere had a particular combination of genes and sequences that just so happened to get into a human and took off.
Here.
As opposed to the avian H5N1s, which tend to sort of get into humans and then...
That's it.
Only because we can't transmit it. That's right.
But pigs are very good at growing all sorts of flu viruses, avian, human, and pigs.
Right.
And they've always been thought to be a mixing vessel.
Right.
And last week, actually, I dispelled this notion.
Yes, you did.
Because there was growing evidence that these bird viruses can go directly into people and they bypass the pig.
Right.
But here we are, a week later. It's clearly a mixing vessel. And,
retracing our steps.
It may be. And one of the problems is that pig farms and poultry farms are often adjacent.
Yes, right.
That's
and the
true.
workers go back and forth, and it's an easy way to spread the viruses. So that may be how you get, in Europe, you got the H1N1 avian virus infecting pigs, and then the people carry the human viruses, and they work on the pigs, and they infect them.
Exactly.
It's really a problem.
Yes.
Right? So that's the origin. It came from a pig. We don't know where this virus came from. We can't say it came from Mexico. We can't say it came from North America.
Nope.
Could have been from anywhere. Someone could have brought it to Mexico and shed the virus, having picked it up on any farm anywhere. And I'm not sure we'll ever learn. Maybe sample collection will give us some clues about viruses that are very close to this. But if you remember, the combination of genes in this virus is a bit unusual. Let me read what Ruben Donis said. He is the chief of molecular virology and vaccines at CDC. It's swine origin. It's almost equidistant to swine viruses from the U.S. and Eurasia. It has close relatives in Asia, and some of the internal genes are from one country, Asia, some are from Europe, North America. So that's the combination that's new, this particular combination of North American and Eurasian swine genes. But remember, the reporter said, so where are the avian and human sequences? And he said, you have to go back 10 years ago in 1998 when the H3N2 human viruses infected pigs and H1N1s and avian H1N1s in Europe, and you got recombination. But they've evolved so long in pigs that they're now pig-like. And that's why we look at the sequence, and if we blast them, all we get are pig sequences. But in fact, the origin is human and avian.
So, Vince, are you going to catch this infection by eating a pork chop?
No, you will not catch this infection.
In fact, now would be a really good time to go out and stock up on pork chops.
That's right. Sausage.
Alan, I thought that the crocodile meat market was going to go up.
Yes, well, yeah. I
tastes just like chicken.
That's what I heard.
There are all these conspiracy theories floating around, and I said, well, who benefits? And obviously it's crocodile farmers.
That's right.
So one thing that may be confusing to a lot of people, this is an H1N1 virus, right? And we have had an H1N1 virus circulating for the past flu season in the world. So why is this able to infect us?
And we've been vaccinating against that too.
Well, maybe we should say how you call a virus an H1 versus an H2 or an H3. What you do is you take antibodies against an H1 virus, and you do an assay and you react them with another virus. And if they don't react, it's not an H1 virus.
Right.
You can do a variety of serological tests. We don't need to go into that. But if the antibodies against one H1 cross-reacts with another, then that's an H1 also. If you have antibodies to an H2 and you add it to an H1, they won't cross-react. So this was done years ago, and there are 16 HAs and some number of neuraminidase genes. So there is, at that kind of test, some cross-reactivity between the H1s, but it's probably not enough to protect you.
Or at least not to protect you completely.
Right. So this is a good question, whether or not we have any kind of protection at all from having had H1N1 in the world.
Precisely.
I mean, you may still get infected, but maybe you have less severe disease.
Well, one of the questions I have is, this country now has a take-up of the flu vaccine of some fairly high percentage. I don't know what it is. In Mexico, they presumably have a lower level, or maybe very low level.
Right.
Have they been experiencing what I would call native H1N1 infections at a high level in previous flu seasons?
I don't know the numbers, but I don't see why they wouldn't.
Not? Yes.
I mean, that was a globally circulating virus for over 10 years.
Right.
Sure.
Right.
That if they had a lower incidence, then that might explain.
Well, I'm thinking about the fact that they're getting deaths among young adults, and whether those people happen to have been people who escaped H1N1 infections in the recent past.
The issue with the fatality rate and the distribution of deaths in Mexico versus anywhere else. I guess it was David Baltimore who said once we shouldn't waste clean thoughts on dirty data.
Yes.
And at this point, I think
epidemiology is largely trying to waste good thoughts on. I mean, I'm not being facetious. I mean, I think that you have to tease out.
Absolutely. I take your point.
Incomplete, fragmentary data, and I think the Mexicans actually did a rather good job because their local doctors said, "Hey guys, we're in the middle of a flu season, which is sort of normal, and all of a sudden we're getting deaths of healthy young adults."
Correct. Right.
And warned people about it, which I thought was, there was an article in the Times today from the former Minister of Health pointing this out, that in fact Mexico did a pretty good job. You compare it with how China did, it's a spectacular job.
Sure.
Yeah.
So I think the Mexican...
my comment was not meant as a slight of the...
I realize that, but
it's just that the data, particularly on the mortality rate, we're dealing with such small numbers at this point, which is good. I hope it stays that way. But it's very hard to say, well, if we actually look at the confirmed deaths, we don't know what the denominator is.
Right.
We don't know
we don't.
how many real infections with this disease.
But the age distribution is unusual.
It is unusual.
That's the point that's unusual.
Right.
Yes.
And so I think that's the thing they picked up, and that's where I think they're to be commended.
Yes.
For latching on to something that if they'd just been unaware, they'd have said, well, this is the flu season, people die.
Sure. I hate to say this because it might deflect our discussion off to something else, but there have been incidences where there were two things going on at the same time, and one thing masked detecting the second thing.
Right.
Yes.
And these death rates may very well be due to two different things.
Sure.
And if that's the case, and the unconfirmed cases turn out to be different, then you've got something else to worry about too.
Right.
This is an important issue because I think it's the lethality, the apparent lethality in Mexico that is scaring people.
Yes, I agree.
And the age group affected.
And the age group. If you take Mexico from the numbers, I don't think there would be as much of a panic.
Not at all.
Because it's only, and we should point out that the strains, so many sequences have now been determined for these strains.
Yes.
We haven't yet seen the Mexican sequences. We don't know why. But this fellow at Ruben Donis at CDC said they're virtually identical to all the other strains.
Right.
He said he's seen 300 different strains. And
got it.
So if we say that they are the same, huge numbers, right?
It's real-time molecular biology.
No, it's amazing. I'm thinking of the people working in the labs cranking out the PCR and the sequencing.
That's right.
It's amazing.
It is.
But anyway, this is an important point.
It's actually one of the reasons the Human Genome Project is paying off.
Hear, hear.
Yeah, all this technology, exactly.
Right.
Good point. But anyway, these are all the same virus, or more or less the same.
I mean, there'll be
there are differences, but mutations that don't account for the difference in lethality. So what accounts for the apparent increase in Mexico? So the one possibility is that the numbers aren't big enough yet to make a determination. In fact, right now...
Besser said that he expected deaths by the time we got, thousands of cases.
Elsewhere. But in Mexico, the lethality right now is 10%, and that's too high for any flu.
Right.
That's right.
Higher than 1918.
Well, that's the suspicion that's due to something else.
So there are other factors at play.
Well, the denominator, I think Alan said the denominator. We don't know the denominator.
Sure.
That's the problem.
That's right.
Right. I've seen in the case reports, you could calculate a 10% mortality rate, or you could calculate a 1% mortality rate, depending on which set of numbers you're looking at.
Exactly.
Sure. But I don't know why we don't have the sequences here to look at.
Yeah, they've released all the others, right?
Yes, all the American, German, New Zealand, Australian sequences, they're released immediately, not the Mexican sequences. I don't know what the motivation is. I've read various things on the internet.
Where are they being sequenced?
In Mexico City,
in Mexico.
sure.
Oh, in Mexico.
The National Laboratory. Okay. Yeah, they have a very good...
But now CDC has received the strains, so presumably they'll do some sequencing. I'll bet by Monday we have...
Well, is it a bit like Indonesia, which refused to...
Tell us about that. What was that all about?
Well, I can't remember the details, but there was, when they were trying to get the collaboration for the avian influenza.
Oh, right. H5N1.
Indonesia was very reluctant to share information because it felt that it was not proprietary, but it could be used by others for economic purposes, like to make a vaccine from which they'd make tons of money, and Indonesia would get zilch.
There's tremendous sensitivity about this in the developing world.
Right.
I have another question for all of you, and that is, if this virus is 10 years old in pigs, and it circulates in pigs, isn't there a hypothesis that suggests that as agents circulate through a given host, they reduce their virulence to become more adjusted by loss of pathogenicity?
Yes, well, pigs don't get very sick from this virus.
No, I understand that, but...
and they've reached a stasis in pigs, right? Pigs are they're not a natural population. They're farmed and then slaughtered every how many months, right?
That's right.
So I don't know, the normal host-virus relationship may not apply. But anyway, they don't make pigs very sick. And I read that many pig farmers ship pigs that are sick because they're not very sick from influenza.
Right.
So I think it's evolved to be happy in the pig. But then the point is, this jumps periodically into people, and most of the time it's a dead end.
Right.
But in a certain person, a certain one virus will squeak past the bottleneck, and that's all it takes. It expands.
Now, of course, the receptor is the same as the human. I mean, the sialic acid
is the human.
So it's not the problem that the avian one has of having a receptor,
that's right.
which is
a little bit different.
Yeah, which is different, so it doesn't infect the upper respiratory or the upper
human.
part of the lower respiratory.
Exactly.
Yeah, these are human. Well, though pigs have receptors for both.
That's right. Yes,
but these strains recognize the human sialic acid configuration. Right. Now, another issue that's been discussed is using antivirals. They're too interesting. One thing, before we go on to the neuraminidase inhibitors, there are these adamantanes, which are the older antivirals. These strains are all resistant to adamantanes.
Really?
So I was wondering about that, and I looked in the literature, and it turns out that if you just, years ago, isolate viruses from pigs, they're spontaneous isolates that are resistant because there's so many different mutants present that some of them are going to be, by chance, resistant to the antiviral. Right. I thought that somehow this indicated that a human resistance gene had
got in, but it's not.
I mean, it could be. You can't rule it out, but it doesn't have to be that.
Right.
So for these, the adamantanes are out, but they are sensitive to the neuraminidase inhibitors, Tamiflu and Relenza. And I understand that these are being deployed from stockpiles and they're being used. So
where are they being used? So for example, there was again in the Times today, there was an article from a fellow who is not a scientist, talking about his response to the epidemic in Mexico City, and going down to pharmacies, and they've run out of Tamiflu, and they've run out of Relenza.
Yeah, sure.
Now, are they prescriptions or are they over the counter in Mexico?
Everything's over the counter in Mexico.
Is everything over the counter?
Yeah, you can buy penicillin over the counter.
Well, I know you can buy antibiotics over the counter, but I wasn't sure about antivirals.
I think
if this is the case, then those two antivirals are going to be useless certainly within my lifetime.
He just said a lot, folks.
Sooner. I hope sooner.
I think maybe by next season they might.
Well,
be
that's
so. I mean, because of the overuse and selection for resistance, of course.
I mean, exactly. These are only two drugs. They vary. They get resistant independently somewhat. Right. There is some cross-resistant, but you get independent, which is good. But it's only two, and as we know from the HIV antiviral situation, you need a lot, at least 3 and maybe 6, so that you can change the 3 when you get resistance to 3. But we don't have that.
Right.
And that's because science hasn't been funded sufficiently for the past 10 years.
Here. That's true.
I mean, there are many targets, and I think there are candidates, but they haven't been brought to market yet. But if we had more, it would be better. So we have these two, and last season Tamiflu resistance was quite high in all over the world. I mean, this resistance.
Well, H5N1 doesn't respond at all to it, right?
So I think this is a problem. These are now sensitive, but absolutely, as soon as you start using it, Tamiflu is an orally taken drug, which is good because you just take your pills. But the other one...
There's an epidemic in Vince's office.
We should talk about transmission of influenza. Let's do that next after antivirals. Okay. Do you know you just expelled 20,000 droplets?
I do know that, but I covered my mouth. Do you notice?
I have a mask here.
The way to do it is this.
No, I'm fine.
We should talk about transmission and how to stop it. But yes, we're going to get resistance. And some people have said, what does that mean in the fall when the virus comes back? And that's something we need to talk about also. But the virus will be resistant for sure. It'll be much more difficult. Yes. Now, hopefully in the fall we have a vaccine.
Right.
Is it realistic to think that we can have a vaccine in sufficient number of doses for the fall?
I don't think so.
Yeah,
I've read that it requires 6 months.
6 months, right.
But that's making an egg-grown vaccine.
Right.
Right.
And this grows in eggs.
All right, so this doesn't grow well, but as you know, you make a high-yielding recombinant with a high-yielding egg strain, which takes a few weeks.
Right.
And then you can grow it in eggs. But it still takes 6 months from start to release.
Right.
And that brings us to October, essentially. So it's a bit late.
Yeah.
Especially if this starts back in September or even late August, which is not unprecedented.
Right.
So what we should do is tell everyone flu has seasonality, even pandemic flu. Right now, in the Northern Hemisphere, the season is just about over. May is pretty rare to see flu, although we're seeing it now.
Right.
Viruses don't follow a calendar.
Right.
And then it goes away in the Northern Hemisphere, comes back in the fall. Right now is the beginning of winter in the Southern Hemisphere, and that's when their flu season is beginning. So my sense of this is that it will peter out in the Northern Hemisphere, move quickly in the Southern Hemisphere, unfortunately, for which there's no vaccine. So there the antivirals will have to be used, and there we will select for resistant variants. But at some point in the fall, if and I think it will come back to the Northern Hemisphere, maybe we'll have a vaccine by October, but it is a bit late. But I don't think that depending on the antivirals is realistic because we are going to have resistance for sure.
Right.
The only mitigating factor, I noticed a paper a few weeks ago in Journal of Virology where in a guinea pig model of transmission, Tamiflu-resistant influenza viruses do not transmit by aerosol. They only transmit by contact.
Oh, isn't that interesting?
And this may be because the virus, with the mutation that it confers resistance to the drug, makes its neuraminidase a little less effective. So it can't get out of the respiratory tract as well.
So the droplets are less.
Isn't that interesting?
So the resistance is evolutionarily costly.
Yes. Yes. Now,
which is always the hope with antibiotic resistance, and people always thought that was going to be the case, but in fact turned out not to be.
Right.
But last season the Tamiflu resistant strains did spread, so I'm not sure how much this applies to people.
Okay.
But there might be some silver lining in the resistance anyway. So I think we need to talk about transmission a bit.
Right.
But before we do that, we need to have a word from our fine friends at Citrix, the makers of GoToMyPC.com. Dick, have you ever heard the technology called remote access?
Well, since I'm a co-host on this show, the answer is yes, I have.
You want to get to your computer from somewhere else, your office computer. Did you think you needed to be a tech expert to figure it out?
I did in the beginning.
Yeah. Well, here's the solution. You need GoToMyPC. It makes remote access really easy. If you want to get to your office computer from anywhere in the world, any other computer, a living room, hotel room in New York, internet cafe in Paris, Mexico City, you just log on to GoToMyPC.com and your office desktop instantly appears. It's built for non-technical people like you, Hamish.
Good. Excellent.
So anyone can use it. Plus, you get free customer service 24/7.
Wow.
You can try GoToMyPC free for 30 days, but you must visit GoToMyPC.com/podcast. That's GoToMyPC.com/podcast. Transmission of influenza.
Aha.
Dick, tell me one way.
Person to person. Achoo.
All right, that's aerosol.
Droplet
Aerosols. Another method.
Ooh. Fomites.
Fomites. Which are objects contaminated with the virus.
Is that a real problem with flu?
That whether it's a problem or not.
Shaking hands. It is.
Yes, I will tell you why. But what's the third method?
Well, fecal oral, but it's
no, it's not fecal. The third method is close contact, kissing and shaking hands, that sort of thing.
Shaking hands.
Now, aerosol, when you talk, sing, breathe, you make aerosols.
Right.
Of course, when you cough or sneeze, you make even more.
Right.
And that expels droplets with viruses in them, just as Dick did a little while ago. That's assumed to be the major route of transmission.
Right.
But contaminated surfaces, daycare centers, studies have been done which show that the toys are contaminated with virus. And here's an interesting one.
And they're viable for a reasonable length of time. So
Hours.
There was one study done. They looked at Swiss banknotes, and they found that infectious virus could last for days, especially if there's mucus on the banknote with the virus. So it could last long enough. So a doorknob, a pole in the subway, windows, bathroom fixtures, they can all have virus. So in a time like this with a lot of virus around, you just need to be careful.
Wash your hands.
You need to wash your hands. You need to use either soap, and remember, you need to wash for the length of the alphabet. Recite the alphabet.
Isn't that a nice little clue?
That's what ASM says.
How nice.
Or you use one of these alcohol-based cleansers.
Right.
More than 60%
Wipes.
Alcohol.
Yep.
So that will help to contact, and but kids are touching each other. It's very difficult. So that's why now there's a federal recommendation. If there's one case of influenza in a school, they close the school for a week.
Yeah.
For a week.
To try and prevent spread.
Right.
Wow.
Right.
Here's an interesting story. It's actually a paper published in the 70s, but it's relevant. It was an airplane about to take off with 75 passengers, and it was delayed on the ground for a few hours, and they turned the ventilation off. Within 72 hours, 75% of the passengers got flu.
Wow.
And there was one person on the plane who was infected sitting up front. And so aerosol transmission, he may have been coughing, it's just breathing, enough to spread the virus throughout the plane and infect everyone. So aerosol transmission is a big deal. So now the recommendations by CDC are if you cough, to cover yourself with a tissue so you don't spray.
Exactly.
Right. Or cough into the crook of your arm.
Which is what I do, yes.
Yeah. Well, it was so severe, the Mexican officials canceled Cinco de Mayo. Right. Which brings everybody to the streets.
Right.
And that was just canceled. When they cancel something like that, they had a soccer match with no audience.
That's right.
So, Alan, what's going on in your daycare center?
Well, they say they're wiping everything down and washing the children's hands repeatedly, and I don't actually have any illusion that would stop the spread of influenza from child to child. I mean, the kids are 3 and 4 years old. They're gonna touch each other,
but of course,
cough on each other.
But of course.
Oh, so we have some CDC PSAs we can put in here.
Yes.
Good, we'll do that.
Health officials are concerned about a new flu virus of swine origin spreading from person to person. To help fight swine flu, cover your nose and mouth with a tissue when you cough or sneeze, then throw the tissue away. Wash your hands often with soap and water, especially after coughing or sneezing. Stay home if you're sick and limit contact with others to keep from infecting them. To learn more, visit www.cdc.gov/swineflu or call 1-800-CDC-INFO. A message from HHS.
I have been blogging a lot about this, and I made the statement that face masks are not recommended by CDC because if you look on their homepage, they say wash your hands, sneeze into a tissue, just use good hygiene. And I got a lot of flack. People sent me articles saying that face masks are effective. Well, yes, in certain settings they are. And let's just say there's a face mask, which is just a flimsy thing like I have here in my hand, which you wear when you're sanding drywall. And then there's a respirator, which clamps over your face with a filter.
Quite different.
Now, I think the reason, this is my guess, that CDC doesn't recommend them is because they know they don't work, that just a face mask won't be used properly. It'll be used loose-fitting, and people will get a false sense of security.
Correct.
And then they'll go out more than they would otherwise.
Exactly.
There you go.
So you see photographs of people in Mexico City on the beach with face masks.
Right. Now, I don't know if that's to prevent them catching it
or giving it.
I think that's a preventative on their part, thinking they won't catch the flu, not that they've actually got the flu and are going out with it.
Well, it seems to me that the air...
Because there's too many. Every second person on the street in Mexico is wearing a face mask.
Yeah, well, you can see that in China a lot, too, though. But I think that's for the air pollution.
Yeah, I think the issue here is the research is usually done in a controlled setting where you have people who know how to put on a mask and who are using one that's either like an N95, the type of thing you'd use to work with a hazardous powdered substance, or an actual respirator like Vince mentioned that has an airtight fitting around your face. And it's not as simple as it looks. You actually have to get this thing fitted correctly, and if it's a respirator, you also have to purge it properly and make sure that you don't have the caps on when you put it on, or you'll suffocate yourself.
Right.
Oh, you're quite right.
So I think the CDC advice is based on
practicality.
People are most likely to go out, buy the cheapest thing that they can find, which is going to be a little surgical mask that's essentially just a glorified handkerchief, and they're going to tie it on their faces and think that they're protected, and in fact not be, and then they're going to have more contact than they would without the mask, because there's a huge body of research that people compensate for risk like this. If you increase safety in one area, they'll decrease safety in another, and this would be disastrous because they wouldn't actually have any real protection, but they would think that they do, and you actually get greater spread. So I think that's where that recommendation comes from.
Right. Yeah, I think it was important for us to talk about that a little because.
So back in 1918, during the height of the spread of flu throughout the world, and a number of quarantine centers, a number of sequestering of large populations of people, did that actually work to stop the epidemic? No.
Well,
do we understand why it stopped?
Surely it's that you get herd immunity.
Well, at a certain point, you get enough immunity in the population, you stop the transmission. You need a certain percentage of susceptible individuals, and once you reach that, it stops.
And isolated communities didn't get it. I was just reading in the flu book by Gina Colata that one of the oddities was to try to actually transmit the flu from sick people to healthy volunteers. There were experiments which were done, which were now completely ethically undoable. A group of Navy prisoners, all of whom had theoretically not had the flu, and flu was raging in Boston at the time, and they were on an island in Boston Harbor, and they got them to try to transmit the flu to these people. And none of them got infected. They took nasal secretions from people dying of the flu.
They were trying to verify the transmission cycle?
They were trying to work out what the transmission was and whether it was a bacterium or whether it was a virus and so on and so forth. And they went to all sorts of measures to get these people infected, and they weren't infectable.
Wow.
So, of course, the caveat here is that it's not clear that they may have had a subclinical, but they would had to have, all of them would have had to have had a subclinical infection prior to the experiment.
Interesting.
So there are some oddities about transmission that I think we don't fully understand. The thought, Gina Colata makes the point that maybe people who are in extremis are not people who are actually secreting virus at the time. So that's probably the likely explanation for this. But it really floored people as to why, they could not transmit this thing.
Do you remember Koch's original attempt to transmit cholera?
I don't remember that, but
it's a very sensitive organism, and it tends to die off if you don't take care of it very well, and to swallow an entire flask's worth of dead organisms.
It doesn't help.
Didn't actually complete his postulates.
Right.
I think it's important to point out that you don't need 100% immunity to stop transmission.
Right.
Yeah.
So for every virus, it's different, but you just need a certain percentage to keep it, and then you fall below that, you susceptibles, and then you don't.
Of course, you look at the plague outbreaks in Europe. The first one killed, half of Europe or a third of Europe, and the next one that came through 100 years later killed off 2%. The next one killed off 0.01% or something. You can see the immunity genes accumulating in the population.
Well, that may also have been evolutionary too. It may have actually HLA types thought to have been affected by the first flu.
Selected out for all the susceptible.
Right.
And then in that same vein, the reason it's spreading has to do with properties of the virus also, but no one really has immunity to this.
Right.
It's a brand new, and immunologically, it's a brand new virus, so it gets in the population and it spreads. And that, of course, is what a epidemic or a pandemic virus is. So this is, it's a little different because I think it is the first time where we have a pandemic strain of the same subtype. Something that's already been circulating.
That's right.
Because we've had H1N1 circulating, but this is a pandemic strain.
We don't know what the pandemics of 1840-something were, or 1898, or whatever the... Are there any old sera?
I have this textbook here.
Written by some
schmo. I don't know.
Racaniello.
All right, 1889.
Yeah, 1889. That's the one.
H2N2.
Oh, so they know that now. Okay.
And 1900 was H3N8.
Okay, so 1900 is. What about? So
that's as far back as I have.
But they are things that have been pandemics before. So the Asian flu was H2N2.
Yes.
Right.
Mm-hmm.
Hong Kong flu was H3N2. Prior pandemics had been H2 and H3. That's the point I'm trying to make. They weren't H6 or.
Right.
But, Hamish, I'll bet the H2 from 1889 was very different
totally
to the
different.
H2, yes.
Yeah, that's right.
If you introduce that into a 1959 population, it would probably be pandemic. Right. Just like today, the swine originated H1 is different enough from the Russian H1 that it's taking off. So I think it's important to point that out. And again, the caveat being there may be some cross-reactivity.
Yes.
I haven't seen the data.
Now, Hamish, I have to point out a lot of listeners are going to be very amused that you initially identified yourself as a layperson when it comes to flu. but the listeners probably don't realize that this is a fairly typical Hamish habit. That his.
No, I genuinely believe this because I've never worked.
No, I believe that you genuinely.
And I don't know. For example, a true flu person would have known those facts.
But yes, but sometimes there are facts and then there's scientific method, right?
Right.
Yes.
When you know the scientific method,
exactly.
you can interpret scientific information because you are a scientist. In fact, you are a virologist, and you worked on a respiratory pathogen. And I have
an anecdote.
You've been trapped.
I have an anecdote. When we used to have labs for the medical students back in the dark ages, when they could do experiments on each other and they could take blood from each other. As an exemplar, I had my blood drawn, fortunately not by a medical student, but by an attending physician, Dave Hodes, who drew my blood very nicely.
Oh, my father-in-law.
Your father-in-law? Oh,
David Hodes?
David Hodes. Oh, isn't that extraordinary? I didn't know that. Well, Dave and I actually have another connection rather than a hypodermic needle. He also was a Rhodes Scholar at the college I went to in Oxford, so that's sort of interesting. So, there's lots of connections.
I'm about to leave the room.
So anyway, they took the serum, and they took the serum of all the students, and they took the serum from me, and we did hemagglutination inhibition experiments, which Vince said we wouldn't go into. But basically what you do is you mix up the serum with a flu sample and see if the antibodies react with the flu sample so that the flu can no longer agglutinate red blood cells.
Correct.
So it's a relatively simple test. You get chicken red blood cells, you get a little bit of the serum, a little bit of the virus, and you do the experiment. And it actually works even in a class of medical students. And interestingly, even though the circulating strain at that time was H3N2, my serum was H1N1. All the students was H3N2, even though I'd had H3N2 fairly recently. And this, I believe, is a phenomenon in influenza virology called immunological memory or immunological recall.
This original antigenic sin.
Original antigenic sin. That's right. That's I knew I was missing the full flavor. And of course, the first influenza that I would have had would have been H1N1 because I was born in 1944.
That's right.
And that was the circulating serotype.
It was.
Ha!
So this was a very interesting exercise for the students because they all had H3N2 and I had H1N1.
It's beautiful. Yeah. Very nice.
Cool.
Oh, it's your father-in-law. How about that?
Well, when you speak to him, say Hamish says hello to Dave. Okay.
So someone asked me, actually Dr. Ginger Campbell of the Brain Science Podcast, who's an emergency room physician in Alabama. Some of the scattered cases don't seem to make sense. Do you think these just represent the fact that we are now looking for it? After all, we do see scattered flu cases in the off-season, but unless the patient has unusual symptoms, we don't typically send specimens to CDC. I think perhaps so. Any respiratory illness now is being flagged for sampling and diagnosis. Yeah.
The scattered cases might have originated from an epicenter in Mexico where they flew back to their respective places, like happened at the school, for instance, in New York. And in Scotland and in other places.
The two Scottish cases had just come back from Mexico.
That's right.
Yeah.
So if you look at if you draw lines back words
like
as to where people have been.
With SARS. I mean, they did a beautiful job with SARS.
I mean, the original map maker, John Snow. Come on, let's go back to this.
Right.
Take that principle and apply it now with Google Earth, and you've got the story.
Google Earth, of course, that we have to talk about Google. Yes. Google has teased out. This is the David Baltimore crappy data. Don't waste good thoughts on crappy data stuff. Google has been looking at the Mexican hits on the internet.
Oh, wow.
And they've been sifting through the data, not just looking at people looking up swine flu, because that's sort of too late. They've been looking up for thermometers and body aches. No, they've been looking up symptoms and people self-diagnosing that they've got something wrong with themselves. And it predates by the same period of time as it did with the CDC when they did that controlled experiment with CDC.
I'll be darned.
So like 3 days earlier. Google was predictive 3 days earlier than the Mexican
health officials.
Actually, there was
amazing
some information on that here from Ruben Donis in his interview.
Yeah, where was that interview, Vince?
It's in Science.
Oh, it's in Science, okay.
So they ask him, what do you think of the outbreak? This is the first one I've seen firsthand as a virologist. The avian flu outbreak is not comparable because this is unfolding so quickly. It reminds me of SARS.
Yeah. Okay.
With avian, there's very little transmission. And even with SARS, transmission was far less.
Right.
Does this one scare you? I saw figures that scare you. We received 300 samples from Mexico, and these cover the span of February, March, and April.
Right.
And you look at flu A, traditionally it's H1 or H3 or it's B up until the end of March. There are 2 or 3 cases up to the last days of March that are swine. Then in April, they skyrocket. So all the cases in Mexico City areas where most samples come from, it really transmits very efficiently.
So it started in March.
So what's the date of the first sample? I think it's the end of March, the first positive. Did Mexico react quickly enough? They didn't know. They probably thought it was regular flu.
Right.
Flu is a seasonal disease that peaks in winter. Maybe this will end in the U.S. We're in a good position. The folks in Buenos Aires are in trouble.
Right.
They're entering winter now. So those are the things we've touched on here today.
Right.
I get quite a few emails on my blog. I thought I would share a couple of them because they actually point out interesting things. They've asked a lot of the things that we've covered, but here's one. I live in Mexico, and we are very confused here about the measures the government is taking against flu. I've heard that it is not possible in the first 7 days to pass the virus person to person, but only in the moment that the symptoms are visible. Is that true? So he's asking, can you shed virus for 7 days before you get sick? You know, flu is a rapidly developing acute infection. Right. So I would say, you can't say never with viruses. There could always be people who shed without symptoms. But I say in most people, the infection incubates about a day or 2 at the most, and maybe you would have a day before symptoms, and you might have virus in that time. But my understanding from volunteer studies is that the symptoms, the virus levels are present about a day before peak symptoms, which makes sense because it's growing, it's incubating. Another one is this is from Mexico also. I live in Baja. This swine flu issue is getting really scary, and I suspect authorities give one information, then give contradictory information. I think they don't want people to panic, as we already are anyway. La Paz is really hot weathered, unfortunately, and the hot weather hasn't come yet. Do you think the hot weather would help to attenuate spread? And of course, we know that
it does.
Hot weather is bad for flu transmission. How long? Yes.
You might mention the Peter Palazzi experiment.
It reduces flu transmission.
Sorry, Alan?
You said hot weather is bad for flu transmission, and I was just clarifying. It means it's bad. It's bad for the virus.
Oh, sorry. Yes.
Right.
Yeah, the virus is
it discourages transmission.
In fact, the Palazzi study in guinea pigs shows that good aerosol transmission requires high humidity and low temperature. Low humidity and low temperature. In the summer, of course, it's humid and hot.
Why is the humidity? I would have thought that was the... that's
well,
curious.
The idea is that the aerosol particles dry out in the winter and travel farther. And when there's high humidity, they absorb water and fall closer to the ground near the person who's coughing. Like Dick.
Right.
Once! I coughed once. Come on.
Oh, I'm just kidding.
One cough doth not an epidemic
How long do you think it would take to make a vaccine?
Okay, twice.
6 months.
Right.
6 months.
Yeah.
Now it is really difficult to find Relenza or Tamiflu, but I have heard that some natural products help reinforce the immune system. Would you recommend an onion?
It certainly would isolate you from the others.
I would highly recommend eating lots of onion and garlic because it will prevent you from getting very close to other
Alan,
people.
I totally agree with that. That's
good.
Dietary quarantine.
Yes.
Now, other emails we get, a lot of people think this is an engineered virus.
Oh, come on.
Mm-hmm.
And they say, well, it just can't be that it's got parts of human and bird and swine. This must be made by someone. It's a conspiracy. And I try and explain how it's impossible for any human to know what kind of virus to make that would spread efficiently in people.
Right.
But somehow I'm not resonating.
Okay.
So could you guys help?
When I see these types of, this comes up with every emerging disease. You see these conspiracy theories. Oh, it must have been created by the government. I mean, first of all, the whole idea that there's a conspiracy, and often it's a conspiracy involving the government. We're talking about our government is going to conspire to do something like this. That's just not credible. In terms of the overall competence level. But beyond that, if you were going to engineer a virus, and this is what you came up with, I'm not impressed. It's not Tamiflu resistant. It's not Relenza resistant. Many of the cases seem relatively mild. If this was engineered by people to be some lethal pandemic, then so far it's not knocking my socks off. So I think there's a real credibility gap in trying to claim that.
Now, is the person suggesting perhaps that, scientists are always making new viruses, and I made my career doing that? Are they thinking that this is something that was made and escaped, rather than a deliberate act of bioterrorism?
This email, they just say, is this a genetically engineered virus?
Well, yeah. So I think that deserves an answer. I mean, clearly, when people are making the vaccine strains, they're genetically engineering by traditional methods. I guess it's still traditional methods, not through. Right. It's not
reverse genetics.
Not reverse genetics. They are engineering viruses. So clearly, scientists do spend time engineering viruses, and they're not omnipotent, so they could make a virus that is, theoretically at least, they could make a virus that would be disadvantageous. And it might escape if conditions weren't appropriate. This could
be
an accidental thing. So I think that there's absolutely no evidence that this is the case. But I think it is something for why we have very high levels of containment and why any breach in those containments is considered to be a disaster. And there have been instances of breaches of containment. So I think it is incumbent on us as scientists to emphasize that whenever we create new life forms, we have to use the precautionary principle, which is that you have to prove they're safe before you release them. So I think that there's no evidence in this case, but it's something about which we have to be open-minded.
That is a very good point.
It's a good point, yeah.
I ridicule the idea that this was maliciously engineered.
Yes,
we can't exclude the possibility that it was.
I don't think Al-Qaeda has got a lab in the desert that's doing this somehow.
It seems pretty unlikely. But this at the same time, this fits all the
conspiracy theorists.
It fits that, but it fits exactly what you'd expect from a naturally arising pandemic. We've got virus that recombined, we now know, not from human and bird and pig. We've got virus that recombined apparently in pigs. And it's very, very easy to believe that happened. In theory, it could have been engineered in a lab, but it just there's no reason to believe that it was.
Also, I think that's a good point because I was looking at it from an intentional, I'm setting out to create a pathogenic virus, and my point is no one knows how to do that.
Right.
You can take existing viruses and release them, but then we know that they are not engineered. But if you wanted to make an influenza from scratch and you had all segments of the various species, how would you know how to put them together? And even if you made one with 8 different segments, it probably would be sick and not spread. So.
And my point was, you would have at least put in antiviral resistance if you were.
That would make sense. Sure.
Yeah.
If you're going to engineer.
In terms of accidental release, I think Hamish makes a very good point. That's an important reminder that we don't have any evidence this was that, but it just reminds us that we need to be careful about containment.
Yeah. My other thought on this is that Vince made this point earlier, and I think this is relevant to this comment. We really should be looking at our farming practices. We really ought to be segregating poultry from pigs.
Right.
Because we are setting ourselves up for this kind of event to happen for the foreseeable future.
Correct.
So the pigs are doing their experiments for us.
Yep.
Well, I don't know if you had, even if you had segregated them, you could still imagine.
Oh, it would still happen.
But
I mean, I think we're making it. I mean, the places like Holland where there are farms right next to each other and veterinarians going from one to the other.
I think we've covered everything. Is there anything else we should discuss that anyone can think of?
Prevention, we did that too.
We did prevention, vaccines, antivirals, where it came from.
Closing
Where
schools.
is it heading in the next week? What do we think is going to happen?
Make some predictions. It's like predicting the college football scores.
Let's make predictions as if we were epidemiologists and virologists, educated.
Right.
Now, I only look at the confirmed cases. Have you looked at suspected, any of you?
Mm-mm.
So we don't know what the trend is. Is it still rising? Is it plateauing?
Suspected cases, it's a little tough because the news outlets have mostly now switched over to trying to focus on the confirmed cases as well.
Good. Well, that's good.
Which is good. But my prediction, which I think is going to be a very safe one, is we will see increases, probably very large increases in the number of confirmed cases.
I agree. I think so.
Because we're looking, not necessarily because it's spreading faster or anything like that, but because we're now intensively looking.
Well, there's some countries that are still relatively free, and South America is relatively free of confirmed cases.
Right.
There are not too many in Australia either. Right. So, I mean, I would think those would be the places where it's going to go. It's the flu season there.
Right.
So I think it will be in those countries in the coming week. And if it's not, then that's quite interesting. What do you think, Dick?
Well, the situation is a lot different now than it was in 1918. Not only are there more people, but there's a higher degree of awareness of what to do when you start to get sick from something. And I think the press has done a wonderful job of alerting the public to the fact that this has the potential for becoming a pandemic, dangerous influenza outbreak, which has caused everybody to go on red alert. Now, when you're on red alert and you're in a country that's gifted in terms of health care, and you can get access to it, then I think the moment somebody gets sick and they're suspected of having this, they're immediately rushed off to a doctor. The doctor takes a look if you get sick or you get to the hospital. Or you stay home. So this sequesters it and localizes it and prevents it from getting to others. And we saw that same thing happen with West Nile virus. The opening shots of West Nile virus in this country were not recognized as West Nile. They didn't know what to do about it. A lot of people died. It had a very high death rate at the beginning, only because of the lack of intervention at the early stages. So, but once they defined what the symptoms were from this, headache, confusion, muscle pain, and malaise, if you had those 4 things and you were admitted to a hospital, you were put on steroids right away. They just assumed West Nile. And the death rate really went down tremendously. I mean, in this country, because of the healthcare system that we have, even though we complain a lot about it, it still works pretty well under these kinds of circumstances. So my prediction, at least for the United States, is that it's going to be localized. The healthcare system will absorb these people. They will be taken care of, and that will stop it because it won't be able to spread. In other countries, like, for instance, south of the border to Mexico, throughout Central America, to places with less well-developed healthcare systems, with rural high populations, with populations also integrated into the agronomy situation with regards to pigs and all these other things, I think you're going to still see a lot of outbreaks, and you'll probably see a lot of spread in those situations. and a lot of deaths because the people are not taken care of as well, and their nutrition isn't as good, and you have a lot of other factors that modify the way these things present. But I don't think we're ever going to have a 1918 again. I really don't think we're going to see it happen like that because of WHO vigilance, international cooperation,
vaccination.
Vaccination.
I think vaccination is going to.
We know what to do.
Helpful.
So
I think the 1918 one had 3 waves, as I recall.
That's right.
And I think it's extremely unlikely that we'll get that.
Totally agree. The only countries that really suffer from things like this, and I'm thinking now of the cholera outbreak in Zimbabwe. Cholera is a very treatable disease. 3 days of oral rehydration, and you don't even need any antibiotics, you're over it. Because the organism doesn't really infect you, it just sort of gives you the toxin, and then it has to burn itself out. But people were dying like crazy in the rural areas because they weren't receiving the proper care. Mm-hmm. So you have to qualify where the flu outbreak is occurring to know what the results will be. And that's what I'm going to say. I'm going to say in enlightened countries with good healthcare systems, this is a totally controllable situation. No need to panic. Be concerned. Don't panic. In high-risk countries with low healthcare capacity, with high populations of poor people, it's the potential for a disaster.
So, Dick, would you not travel now?
Of course not. I would travel.
No,
sure.
There's no risk to traveling.
I'm not afraid to travel anyway. There's a risk of getting into an airplane. But, I mean, am I afraid of catching the flu is what you're asking me.
Yes, of course.
Actually, all my life I have personally avoided taking the flu vaccines only because I was under the impression that you can't predict what this year's flu is going to be based on last year's epidemic. You can make a vaccine based on last year's epidemic, but you're never going to be sure.
Well, some years are pretty good, though.
No, I agree with you. So one year I did take it, and lo and behold, I didn't catch flu either. In fact, I've never caught flu, so maybe I'm one of those rare people
that could never... But in general, for people, we've had people asking, should I be traveling?
It depends. I mean, I wouldn't go to. I don't think you can go to Mexico City right now.
What about Europe?
Well, everything's closed in Mexico.
That's right,
now.
And that's
why
what I'm
I
saying.
Wouldn't go there.
The air traffic controllers are not there.
As far as I can tell, everywhere else it's equal incidence so far.
I would go.
So I would go.
Sure.
We have a couple of picks as usual because we want to have a little sense of normalcy. There's not a panic here.
That's right.
But before we discuss that, let's have a word from the American Society for Microbiology.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Our science blog of the week is Dr. Harley Rothbart's Germ Blog. The inside story on germ defense, from antibiotics and vaccines to home remedies and health food. Now, the reason I picked this is because Dr. Rothbart, in fact, used to work on enteroviruses. He used to be, he's an MD who used to do some research.
Uh-huh.
And I knew him from my days on polio. And actually, I called him a few weeks ago and said, hey, I just found you on the internet. Come to TWiV. And he said, well, I don't do research anymore. I do outreach. So he travels around the country telling people how to prevent getting infections.
Uh-huh.
So he has a good blog where in fact, this week he's talking about how to prevent transmission and getting flu. So I thought that would be a good pick. And maybe we'll have him on a TWiV because he said, if I can contribute anything, I'd be happy to come. So maybe he should come and talk about prevention.
You betcha.
Our podcast of the week is Science in the City, a podcast.
Science in the City.
That's from
I like it.
New York Academy of Sciences.
Yes. Interviews, conversations, and lectures by noted scientists and authors. You must be there. You're a noted scientist.
I don't think I'm on that. No, I don't think I made that one.
And our science book of the week, we've picked in the past the great influenza, we've picked flu.
That's right.
So you've already read those, but I thought it would be good to read The Swine Flu Affair, Decision Making on a Slippery Disease. And this is the 1976, which I think is instructive to look at.
Yes, it is.
And in fact, as Alan has pointed out, you can find it online for free, and we'll put the link to that in the show notes, so that might make some good reading. And
you go to that page, you scroll down if you want to read it for free. The download, it says not available for sale, and of course they don't have it as a downloadable PDF because the thing came out in 1976, but you can read scans of it if you scroll down the page.
Okay, great. That's great. So don't forget, you can find other science podcasts at sciencepodcasters.org and promednetwork.com. A few closing things. Remember, we're going to be at the ASM General Meeting on May 19th, and I guess we'll still be talking about swine, or I shouldn't call it
H1N1.
That's right.
The current H1N1 strain, possibly the pandemic strain.
Yeah.
And we'll keep score. We'll see who actually made the most cogent guess as to what's going to happen.
Notice I didn't guess.
That's good. Hamish has always been a conservative
and one abstention.
You can follow us on Twitter, Alan Dove, PROFVRR. I've been blogging a lot at virology.ws on this, and running a little virology course on influenza if you want to go over there and learn something. Started to do some teaching because I realized that people's questions reflect they don't understand just the basic biology of influenza.
Yeah. Did we finish our prevention by saying that you can't catch it by eating pork?
Yes.
Good. Let's just say that again for one more time.
Pigs are out of the equation now. Leave the pigs alone.
That's
it's
right.
now a human infection, a human
Well,
virus.
don't leave them alone. You should still eat them.
Well, yes, you don't need to slaughter them. That's what I mean.
You don't need to slaughter them.
I think we just lost our vegetarian audience.
By the way, least you think that Egypt will suffer greatly from this loss of pigs, just remember that if you start from scratch, you can raise an adult pig in 6 months.
Mm-hmm.
It's a very efficient way of raising meat.
Why does a Muslim country have pigs?
Because they have
a
Coptic Christians. They have 6 million Coptic Christians living inside Cairo. That's not just a Muslim country.
I know.
And they have tourism. And when you go to Egypt and you see the pyramids, maybe you want a nice Italian meal afterwards, with some sausage and some pasta.
I think when you go to Egypt, you have to do as the Egyptians do. You don't need to eat pork.
No, that's not. No, but 6 million. 6 million Coptic Christians live in the city limits.
What percentage of the population is that? About 10%?
It's about 10%, something like that. They are an underprivileged class of people in Egypt, unfortunately.
I would guess there's some clash between them and
there is, of course, there is.
In terms of pork, for
example, there is.
Right?
There is.
But this is not This Week in Politics, so.
No, it's not. But
This Week in Virology.
There is some politics in virology.
Which, in fact, is what you've been listening to. This Week in Virology, the podcast about viruses, and we're right in the middle of a possible pandemic, H1N1. And the official name, by the way, Mexico City, 2009 brackets H1N1.
Right.
Actually, there's a isolate number in there, and then if it's from Texas, you would put Texas in, or California, or Regensburg, or Auckland, or whatever.
Or Norwalk, Ohio.
That's right. With flu, you don't have a choice about the locale. It has to be in the strain name.
Right.
But, of course, the virus is influenza virus, whereas norovirus is a different story. But these are just isolate names. Thank you for coming again, Hamish.
It was a pleasure seeing you again.
Nice to see you again. It's a nice time to be on TWiV, right?
Yeah, perfect.
Perfect timing.
That's right. That's right.
We'll be back again next week, right, Dick?
Right.
Alan, thank you.
I'll be here.
Bye, Alan.
Alan, you still there? Yeah.
See you, Alan.
Yes, I'm still here. It was good talking to all of you.
We will see if we have other viruses to talk about next week or whether the story dominates. We'll see, but come back next week because we'll be here. We'll be back next week. Thanks for joining us. Another TWiV is viral.